हेलो विरानिका मैम। हेलो
Hello.
डॉक्टर विरानिका, आर यू देयर?
Yes, I am here. I am here.
ओके मैम, स्टार्ट कर दीजिए अपनी पीपीटी शेयर करके मैम।
मैं स्टार्ट कर दूं क्या?
यस मैम।
Are my slides visible? Are my slides visible?
यस मैम।
Okay. Hi, good evening, everyone. And today I have a great pleasure in welcoming all of you to the really exciting world of medical geneticists. So now, my topic here today is that why is genetics important for a pediatrician, and why should a pediatrician think genetics, and when? So we should think genetics when, as a pediatrician, when we are faced with developmental delays, congenital abnormalities, facial dysmorphisms, that is, a face that does not look normal to you. Now, we do not use the term dysmorphism really, we use the term distinctive facial features. So, not as to really label a person abnormal. Then, when there is severe hypotonia and you cannot think of a pediatric or a general infective, etc., cause, unexplained anemia, growth abnormalities, recurrent metabolic disease, and a disease which follows a multi system pattern, and we do not know the cause. So, now we need to understand that common genetic disorders include spinomuscular atrophy. Spinomuscular atrophy is one disorder which is caused by deletion in the SMN1 gene. How and when do you think of this? You think when you find a child with tongue fasciculation, floppy infant, floppy child with areflexia, always think of SMA. And what do you do when you think of SMA? You order a genetic test for SMA. Now, here is a 5-month-old infant with progressive hypotonia, poor head control, even at 5 months, weak cry, reduced reflexes, and tongue fasciculations. So now, in such a child, we think of genetic disorders. Genetic disorders are those disorders which are caused due to aberrations or mutations or deletions in a gene. What is really a gene? Gene is what our DNA makes. A DNA makes a gene makes something known as RNA, and RNA makes protein. Now, this is known as the central dogma of molecular biology. That is, a DNA leads to formation of a protein. Now, a DNA is what a gene consists of. That is a genotype, but when we have a protein and a manifestation is caused, that is known as a phenotype. So now phenotype guides really how a mutation in a gene is interpreted. We have chromosomes. There are 46 pairs of chromosome in each of our cells, 22 pairs of autosomes and 1 pair of sex chromosomes. These can lead to something called as numerical abnormalities. One of the chromosomes may be missing, one of the chromosome may be extra. This extra or missing chromosome is what makes an aneuploidy. These are numerical problems of the chromosomes. Now, what are structural abnormalities of the chromosomes? When there are deletions or duplications, translocations or inversions, such things make a structural abnormalities. When you want to think of genetic disorders, you can think genetic disorders in these following headings, that is chromosomal disorders, monogenic disorders, that is the ones caused due to genes. Chromosomal disorders will be caused due to chromosomes. Chromosomes can be deleted, they can be extra. Chromosomal disorders can be detected on the basis of testing that includes chromosomal microarray, and monogenic disorders can be detected by a technique known as next generation sequencing. Now, I'd be happy to know if, like, any of you have ordered a test that is the next generation sequencing testing. So if you have, you can really let me know. And if there are any questions related to that, I'd be happy to answer. The other group of disorders that exist are the triplet repeat expansions. There are certain genes in which some triplet repeats are present which cause the disease. The prototypical example of such a disease is Fragile X syndrome. Fragile X syndrome is the most common cause of inherited X-linked intellectual disability that is present in the boys. So Fragile X syndrome needs to be, okay, someone said that chat is disabled. So I think you can just put your questions in the question answer. So, Fragile X syndrome is the most common cause of X-linked inherited intellectual disability. And how do we check for Fragile X syndrome? By a test known as triplet primed PCR or a TP-PCR. So, going through this again, for chromosomal disorders, you do a test known as chromosomal microarray. For monogenic disorders, you do a test known as NGS, next generation sequencing. And for triplet repeat disorders, you do a test that is known as TP-PCR. Now we come on to mitochondrial disorder. Mitochondria is the powerhouse of the cell, and any mutations or deletions in the mitochondria leads to mitochondrial disorders. So what test we need to do for mitochondria include the sequencing of the mitochondrial genome, and this test is known as mitochondrial genome sequencing. So these are the basic category for genetic disorders. Now, some disorders are not purely genetic, but they may be clustered in the family. These include multifactorial disorders like diabetes, hypertension, clubfoot, neural tube defects, etc. These disorders, when they are present, cannot be detected by one single genetic test. They have small effects of various genes that lead to these disorders, and we do not offer a genetic testing for most of these disorders, except when there is a very strong family history. So, important next is to understand the patterns of inheritance in genetic defects. Understanding a pattern of inheritance can be best done by something known as a pedigree analysis. Pedigree analysis is a graphical representation of all genetic disorders or all problems in a family. It helps you. So this class is beyond pedigree analysis because my topic today is common genetic disorders, but I'll just give you a brief overview of a pedigree, what a pedigree is. When you draw the different affected and unaffected members in a family, that is what you call a pedigree. You will represent males and females in different symbols. So it helps to predict the risk of recurrence. It also helps to identify if there are any relatives which are at risk of genetic disorders and guides genetic counseling. Now, the patterns of inheritance can be autosomal dominant. That is, out of the two genes that we have, only one is affected, the other is fine. So there is a 50-50% chance of the mutated genes to go to the next pregnancy and the next child. So there is a 50% chance for each pregnancy. Now, the classical examples of autosomal dominant disorders I have listed, that is neurofibromatosis, Marfan syndrome, achondroplasia, and tuberous sclerosis. This is a classical example when we have a tall adolescent with adiponodactyly, pectus deformity, and ectopia lentis, and a family history of certain cardiac death. From this case, it is very clear that the child has Marfan syndrome. We will order a next generation sequencing to look at this gene, and the genetic pearl in such a case is an ectopia lentis, a tall stature, a family history of cardiac death, basically showing that there is aortic involvement. So this is an autosomal dominant disorder. The child of the person affected with Marfan syndrome will have a 50% chance of being affected with the same. Next, we have the autosomal recessive inheritance. This is one of the more common inheritance we see. And this inheritance has to specifically be borne in mind because in this inheritance, the person who is carrying the gene may be normal, and such a person is known as carriers. When both the couples are carriers, their children have a 25% chance of being affected. So in this case, there is no vertical transmission, but there is a horizontal transmission. That means multiple siblings may be affected of the same disease, and you may not see many generations affected of the disease. These disorders are more common in consanguineous marriages and more common in marriages where there is marriage between the community. There is a 25% risk in each pregnancy. There is a horizontal pattern when there is sibship rather than the vertical pattern as in autosomal dominant disorders. The classical example here is thalassemia. Thalassemia is a classical prototype example of autosomal recessive disorders. Now, again, we have a consanguineous couple, that is, the cousins married each other, and there were two children with progressive muscular weakness. The parents were clinically normal. So, in such a case, as I told you, we think of common disorders first, think of SMA first. SMA is a disorder that is characterized and tested by MLPA testing, which looks at deletion in the in the SMN1 gene. Now, we also can tell them that in their next pregnancy, there will be a 25% chance of having the same disease, and also explain the reproductive options to the parents. Next, coming on to the X-linked inheritance. X-linked inheritance can be X-linked recessive inheritance. That is, we have two X chromosomes for the females, and for the males, we only have one X chromosome. So females can be normal if they have mutation on the X chromosome, but male, because the other X is working, but the males have a disease when they have the mutation on the X chromosome. So such a disease is only present in males. There is no male to male transmission because a male will only give Y but not X to the next male of the next generation. Mothers and sisters are carriers, that is, the girls are carriers. They will not themselves have a disease because they have two X chromosomes and their sons are affected. Now let's come on to an example of an X-linked inheritance. The boys who couldn't climb the stairs. So a 4-year-old boy had frequent falls with Gower signs positive, a calf hypertrophy, and markedly elevated CPK. Now this markedly elevated CPK is a prototype example of Duchenne's muscular dystrophy. It's an X-linked condition which is found in boys only. And the girls are carriers because they have two X chromosomes. Next is the mitochondrial inheritance. Mitochondrial is a maternal or matrilineal inheritance. It is characterized by heteroplasmy, that is, the load of the mitochondrial is different in different parts of the body, in different tissues of the body, and also the load changes when it goes from one generation to another. So that is mitochondrial inheritance. An example of mitochondrial inheritance is MELAS and LON. And the other inheritance is de novo inheritance. De novo inheritance means the mutation is first time produced in a person and it is actually not inherited from the previous generation. Achondroplasia is a classical classical example of a de novo inheritance, that is mother, father are normal, but the child has a de novo or a new mutation. Now let's come on to chromosomal disorders. So we have dealt with Mendelian disorders. Mendelian disorders are the ones which are characterized by a dominant recessive X-linked or mitochondrial inheritance. Now we have chromosomal disorders. So, when do you think of chromosomal disorders? Again, multiple family members can be affected, but they all may have different features. So, you will think of them when there are multiple congenital abnormalities, developmental delays, congenital heart diseases, dysmorphism, growth abnormalities, failure to thrive, or overgrowth, or congenital heart disease. Also, in genital abnormalities and autism. In all of the conditions, remember, you need to first suspect chromosomal disorders. So now the most common disorder that we need to and always want to talk about is Down syndrome. Down syndrome is by far the commonest genetic disorder. I will be happy if any one of you could tell me that why is Down syndrome such a common disorder. I'll give all of you like a minute to think. Anyone could tell me? I will ask the question again. Why is Down syndrome the commonest genetic disorder? And is it, do all of you think it is a common genetic disorder? Okay, someone said that delayed marriage and childbearing, but there are other disorders which are also caused due to delayed marriage and childbearing, like trisomy 18 and trisomy 13. Why is Down syndrome even commoner than them? Something related to the size of that chromosome, so if you can think about it. Think about how big or small that chromosome is. So which chromosome you think is the smallest chromosome? Anyone can answer, which do you think is the smallest chromosome? सबसे छोटा क्रोमोसोम कौन सा है? 22 ऑटोजोम्स हैं, एक एक्स क्रोमोसोम और एक वाई क्रोमोसोम है, तो ये जो ऑटोजोम्स हैं 22 इनमें से सबसे छोटा कौन सा है? Why? Okay. Autosomes may I want to know which is the smallest. Y is the correct answer. But if we only talk about autosomes, okay, no. So the smallest chromosome is chromosome number 21. Generally, chromosomes that are arranged when we say from 1 to 22, they are in the descending order of their size. Okay. So the chromosome 1 is the largest, but 22 is not the smallest. Chromosome number 21 is the smallest. तो अब कोई बता सकता है that why is Down syndrome the commonest genetic disorder? Okay, so no. So chromosome number 21 is the smallest chromosome. उसका मतलब है उस पे सबसे कम जीन्स हैं. तो अगर उसका इम्बैलेंस है या उसका एक्स्ट्रा जीन्स है तो द पर्सन रिमेन्स द मोस्ट नॉर्मल. If there is trisomy of 20, or if there is trisomy of 18, if there is trisomy of 13, is there is trisomy of chromosome 1 2 3, any chromosome, ये लोग इतने बड़े बड़े chromosomes हैं और इस पे इतनी सारी genes हैं that most of them don't survive. They are such severe disorders because they have so much imbalance due to a complete extra chromosome. Now, because Down syndrome is, because chromosome number 21 is so small, the number of genes on chromosome number 21 are so small, so less. Because of that, imbalance or a duplication of that causes less imbalance and less abnormality, and these children survive. So, that is the reason that these children exist. They get born, they have almost normal lives, but the other chromosomes are not like that. Okay. Okay, so we will now proceed. The baby with severe abnormalities. Let's talk a little bit about trisomy 13, that is Patau syndrome. So Patau syndrome is a severe disorder characterized by midline defects, polydactyly, severe congenital heart disease, and there is severe growth restriction. Okay, and what the test you do is a karyotype or a fish test. So now let's talk about at a glance about common chromosomal disorders. So trisomy 18 is characterized by clenched fist. There is an overlap between the first and the fourth fingers. Like this, you will find the hands of the fetuses with trisomy 18. And there are also severe congenital heart defects that are found. These are disorders are lethal in infancy if they actually reach infancy. So now amongst autosomes, we know that trisomy 21 is the most common disorder, but among sex chromosomes, one of the common disorders is Turner syndrome, characterized by short stature, webbed neck, streaked gonads, and the diagnosis is confirmed by karyotype. So girls with short stature, primary amenorrhea, you think of Turner syndrome. Klinefelter syndrome is the most common sexually inherited sexual disorder of chromosomes. So this is characterized by tall stature. There's an extra X chromosome, gynecomastia, infertility, and generally diagnosed at puberty when these children fail to achieve puberty or make sperms characterized by infertility. And the test you do is a karyotype analysis where you, instead of 1X and 1Y, you find 2X and 1Y. Then we have 22q microdeletion, that is DiGeorge syndrome, characterized by cardiac abnormalities, cleft lip, palate, hypocalcemias. Again, some of them have mild to moderate intellectual disability and diagnosed by chromosomal microarray. Williams syndrome, another common chromosomal disorders in which you will find the children to be very happy, happy puppets. They have a friendly personality, elfin facies. They also have a supravalvular aortic stenosis at times. And one of the easier disorders to diagnose, and the test of choice will be a fish for targeted testing or a chromosomal microarray. Now, this is how a case looks like. A child who comes with conotruncal cardiac defect, cleft lip, velopharyngeal abnormality, hypocalcemia, recurrent infections. There is immunodeficiency. You think of DiGeorge syndrome, and the test you do is a chromosomal microarray. If there is any question about chromosomal disorders, I'll be happy to answer. So we can just take a 30-second break and you can ask some question, because then now we will come on to the monogenic disorders. Okay, so I think there are no more questions. Okay, this is the one question. I think please tell which types it. Which test? So I think I told that already. So when you do have a chromosomal disorder, the best test to do, which will pick up most of the chromosomal disorder, is your chromosomal microarray. Then when you're thinking of a single gene disorder, that is the one with autosomal in or dominant recessive X-linked inheritance, then you will do a exome sequencing. So that is the those are the two basic tests. So in autism, developmental delay, genital abnormalities, and growth abnormalities, you think of chromosomal disorders first. But in single gene abnormalities like deafness, blindness, etc., you think of single gene disorder first. So for single gene, you do an exome sequencing. For chromosomal, you do a chromosomal microarray. So I think these are the basic two tests you remember. There are some disorders which are not picked up by these two. For that, what you need to do is, for Fragile X, you need to do Fragile X TP-PCR, that is Fragile X triplet prime PCR. Then for SMA, you need to do SMA MLPA. Then for DMD, you need to do DMD MLPA. So these are the basic tests that if you know, then that's fine. Any other question? So now let's move on to the next group of disorders. That is, we have a monogenic disorders that are Marfan is chromosomal microarray. No, Marfan is a single gene disorders. All the autosomal dominant, all the autosomal recessive disorders, X-linked, all of these fall into single gene. And chromosomal disorders are when we have only problems in the chromosomes. Marfan is caused by mutation in a gene, not deletion or an aneuploidy. So for that, you do a exome sequencing. Okay. Any other question? Okay. Now we have almost come on to covered more than half of the presentation. Let's move forward. The monogenic disorders, when do you think of monogenic disorders? When we have an inheritance pattern, we have a molecular diagnosis, we need to do a genotype-phenotype correlation, and finally the counseling. Then you have child with severe anemia, that is chronic pallor, failure to thrive, increased spleen size, hepatosplenomegaly, you mostly think of thalassemia, and for that, you need to do sequencing of the HBB gene. So what is this thalassemia? Let's talk of thalassemia a little bit because it is one of the commonest autosomal recessive genetic disorder. Am I audible?
Yes, ma'am, you are.
I think there is some problem. 1 second. Something is happening. Gauri, there's some question I'm not able to open. हाँ, इंटरनेट आ रहा है ना? Okay. अटक गया. If I'm audible, I'll just proceed. Gauraji, I'm audible, right?
Yes, yes, ma'am, you are audible.
ठीक है, I'll just proceed. So now let's talk about DMD. So Duchenne muscular dystrophy is an X-linked disorder. It is characterized by X-linked inheritance, very high CPK, and the diagnosis is made by genetic testing of the DMD gene by multiplex ligand probe amplification. These are boys who present after 3 to 4 years of age with difficulty in walking, pseudohypertrophy of calf, and progressive difficulty in climbing stairs. We now have multiple treatment options, but definitely the prevention in this case is better than the treatment options available at present. Care is steroid therapy, but we now also have exon skipping, gene therapy, etc. So now the common monogenic disorders include, when we have central hypotonia, we have disorders like brain-based origin, in which there is a brain-based origin. HIE reflexes are generally hyperreflexia, or at least preserved. They can be present in leukodystrophies, neurometabolic disorders, or CNS, metabolic CNS diseases. Peripheral hypotonia, this is reflexes are reduced or absent, type SMA, DMD, and metabolic. Some of the metabolic disorders also have peripheral hypotonia. SMA we have discussed, so I'm going to skip that. Facial features, then we have to actually consider both testing, because it is difficult to make a diagnosis. Sometimes we can look at the child and make a diagnosis, but if you cannot make a diagnosis just by looking, you can order both exome and add it together. Like I spoke about, it is the most common inherited cause of X-linked ID, and it is caused by mutations in the FMR1 gene, and it is caused by expansion of the triplet repeats that are present. It is characterized by intellectual disability, autism, Hello. Yes. So sorry, I got disconnected. Someone else will be sharing my screen. So about thalassemia, yes, thalassemia is also a single gene disorder, but because it is such a common disorder, we can sequence that gene separately as the gene is available separately for sequencing because it's so common. Otherwise, it will also be picked up. You're right on clinical exome. So you can do exome sequencing for all single gene disorders. So that is correctly asked. That is thalassemia can be picked up on exome, but you can. It is better to do an HBB gene sequencing for the same as well. So I think we have covered quite a few disorders in detail, and there are some 7 or 8 slides left. Once they're connected, we can discuss those. And if there are any other questions, then you can ask. Otherwise, I would like to just summarize the talk that genetic disorders need to be remembered in pediatrics, especially when you are looking at an infant with global developmental delay, that is, developmental delay in 2 or more milestones. When you're looking at autism spectrum disorder, a child with failure to thrive, overgrowth syndrome, or dysmorphic facial features. So, in all of these cases, do think of and tone abnormalities, always think of a genetic disorder. Now, genetic disorders are majorly of two kinds, chromosomal and single gene. Chromosomal disorders consist of manifestations of various systems. So, when there is a multisystemic involvement, multiple congenital abnormalities, we think of chromosomal disorders. And when there is abnormality of one system, when there is a neurological involvement or a deafness, blindness, tall stature, short stature, you first think of a single gene disorder or a hepatosplenomegaly, etc. Now, single gene disorders are tested by next generation sequencing. Two major tests of next generation sequencing include two major tests of next generation exome sequencing and clinical exome sequencing. Exome sequencing looks at all genes together. Please go to slide number 39. Yeah. So now, so this is a group of disorders that we have not touched upon, and this is the final few slides. So the child who becomes sick after feed, that is a classic metabolic disorder. The child was normal at birth, but there was a vomiting after protein intake, there was altered sensorium, and ammonia's levels were high. This is a characteristic of urea cycle or an organic acid urea. These disorders are typically treatable, should be picked up early and treated early as well. In such disorders, metabolic disorders are all single gene, and you can do an exome sequencing for these. Next slide, please. So, inborn errors of metabolism include amino acid disorders, for which you do a UPLC of amino acid and a tandem mass spectrometry, organic acidureas, for which you do a urine GCMS, urea cycle disorders, for which you do urea cycle disorders, for which you do again a TMS, fatty acid oxidation defect, for which you do a TMS, lysosomal storage disorders. Some of them have and enzymes which are available. If enzymes are not available, for example, we have beta glucosidase in Gaucher, alpha glucosidase in Pompe. But if we do not have those enzymes, then you can do an exome sequencing. Peroxisomal disorders are characterized by a metabolic test called VLCFA. In short, all of them are single gene and you can do exome for all. Next slide, please. So this is an example of a patient with developmental regression, one of the hallmark cases of genetic disorders, and any regression is abnormal and has to be investigated. You think of metabolic disorder, a neurodegenerative diseases, epileptic encephalopathy, or a syndromic diagnosis. Regression after a normal developmental trajectory is a red flag that has to be investigated, and it broadens actually and does not narrow the differential. MRI brain is the most important investigation, followed by exome sequencing again. Next slide, please. So, choosing the genetic test is one of the most challenging parts in genetics. There are all the variety of tests that have been mentioned in this slide, and you can take a picture of it as well. Next slide, please. So, this is where I give you a little brief of the different genetic tests. If you have Down's phenotype, you do a karyotype. If you have a DMD phenotype, you do DMD, MLP, or developmental delay or dysmorphism, do a CMA first, exome second, a suspected fragile X, you do FMR1 gene testing, that is triplet repeat PCR. If you have a metabolic disorder, you do metabolic testing and exome sequencing. Next slide, please. So, when should you refer to a clinical geneticist? Geneticists should be involved in decision making in presence of these different things that I have mentioned on this slide. Multiple abnormalities, facial dysmorphism, progressive weakness, you do not know the cause, unexplained infant death, that is recurrent sudden infant death syndrome, multiple abnormalities of the organ, familial disease, recurrent pregnancy losses, suspected metabolic disorders, and the most important is developmental delay or intellectual disability. Next slide, please. So, finally, I would like to put all of these together. There is a child with developmental delay, dysmorphic features, congenital heart disease, hypotonia, and short stature. You need to know, first ask that if this is genetic, if it is chromosomal or monogenic, or what test you want to order. But the Noonan syndrome here is a diagnosis of a single gene. Why we want to look at Noonan syndrome? Because the developmental delay was not so severe. But if it was, you could have ordered a chromosomal microarray. So when you have dysmorphism and short stature together, it is better to order both tests together. Next slide, please. So now, it has been a privilege to interact with all of you, and I would like to just end by saying that think genetics when you think of congenital unusual manifestations. There is a family history. The disorder is progressive. It is unexplained. Everything unexplained. You should think of genetics. Always look at the child in detail. You will find small clues like neurocutaneous markers. Each pedigree inheritance pattern is very important. Choose the right test for the clinical question. And remember that a negative family history does not exclude the genetic disorder whatsoever. An early diagnosis now in so many myriad of treatable genetic disorder helps in treatment. If not treatment, then surveillance and recurrence risk counseling. Thank you so much. Thank you. Thank you, Gaurav ji. Can I leave now?